Hi there, welcome to this podcast on meiosis. The objectives of the podcast are to answer what is meiosis, how does meiosis make 4 daughter cells, and how does meiosis contribute to genetic variation. Meiosis. It's a type of cell division in sexually reproducing organisms. That's the majority of organisms, except for a few plants that only reproduce asexually. Starts the diploid cell. Notice the DI sound in the diploid? Well, that's referring to 2. So, in diploid cells, the chromosomes are in pairs. In the case of humans, there are 23 pairs of chromosomes, so giving us a total of 46 chromosomes. An example of a type of cell that's going to carry out meiosis are stem cells in the testes and also in the ovaries. What's interesting is previously scientists believed when a female was born, all of the eggs that she needed throughout her life had already been stemmed from cells inside of her ovaries whilst she was still in the womb. Researchers now aren't too sure of this, and some of them believe that there are stem cells in the ovaries, so a woman during her life is able to make new eggs or ovum. And the meiosis process leads to the creation of 4 daughter cells, and these daughter cells are the gametes, so the sperm and the egg. These cells are haploid. What that means is they have half the amount of genetic variation. So whereas in the diploid cell, the chromosomes are in pairs, in the haploid, the chromosomes aren't in pairs, so in the case of humans, there'd be 23 different chromosomes. Okay, meiosis starts with a cell. This is a very simple cell containing only 4 chromosomes. It's a homologous pair, so we have 1 chromosome in this rod-shaped structure, inherited from a mother and the other from the father. These are homologous because they are identical in terms of the structure. They are near enough the same length, they have the same types of genes. However, the genetic information between them varies a lot. So for example, this may have a gene that codes for blue eyes, and this may have a gene that codes for brown eyes. Okay, meiosis starts like mitosis, and the DNA replicates. So effectively, the amount of DNA is doubled during DNA replication. The chromosomes have changed from this rod-like structure to this X-like structure, which is 2 identical sister chromatids joined in the middle at the centrome. And then there's meiosis 1. Meiosis 1 is a complete division of this cell. However, unlike mitosis, the entire X-shaped chromosome goes into a new cell, and they go into different cells. So we've changed here from a diploid cell which had 46 chromosomes to a haploid cell, because this cell, although it's got the same amount of DNA as this cell at the start, it only has 23 chromosomes, but these chromosomes you can see look structurally different. You notice too the 2 sister chromatids, although practically identical, have changed a little bit. We see there's a blue tip, a red tip on these chromosomes. That's a process we're going to look at a little bit later in detail, called crossing over. However, simply put, crossing over occurs when these chromosomes align at the equator of the cell before meiosis 1, and the chromosomes kind of intertwine with each other, and when they're pulled apart into 2 new nuclei, genes in the same position can kind of be pulled from one chromosome to the other, so they exchange their genes. So this is one kind of one kind of way meiosis causes genetic variation. In meiosis 2, the cells again divide. The amount of cells is doubled, so we have 4 daughter cells. And these daughter cells are like the gametes, so this is maybe 4 different sperm with different kind of genetic combination. The way this differs from meiosis 1 is that you can see the structure of the chromosome has gone from this X shape of 2 sister chromatids to this rod-shaped kind of single chromosome. Again, this is a stage where lots of different genetic combinations can occur because it's random where these chromosomes go. So it may have just been just as easy for this chromosome to have ended up in this cell and this chromosome to have ended up in this cell. Again, these are diploid, these are haploid cells, and they differ from this diploid cell in that you can see they only have half the number of chromosomes, and in meiosis 1 you can see half the number of chromosomes. If we go back a stage, we talk about independent segregation, and similarly this chromosome may have ended up in this cell and this chromosome may have ended up in this cell. So there's lots of different combinations of genes in the cells during meiosis 1 and meiosis 2. If you think that the human's got 23 different chromosomes, then we can calculate, because they're in pairs, that 2 to the power of 23 leads to an answer over 8 million. So there are over 8 million different combinations of genes in the cells in meiosis 1 alone. Okay, let's look at some additional resources. Please go on the BBC Bitesize website and read through the information on there and look at the pictures on meiosis. And then there's a very good information that's from McGraw-Hill, and they, although it goes into a bit more information than we really need at this stage, the animations on there are very good, and so is the commentary, so it's well worth a watch.